Welcome to one of the most fascinating and crucial topics in all of biology, programmed cell death, commonly known as apoptosis. Imagine a city where old, damaged, or dangerous buildings need to be removed to make way for new ones. If you just blow up the building randomly, debris will fly everywhere, causing a massive emergency. But a professional demolition crew carefully dismantles it from the inside out. Today, we will explore how our cells perform this beautiful, orderly process for the greater good of the entire organism. To begin our journey, we must understand the difference between accidental and intentional cell death. In the human body, random, messy cell death is called necrosis, and it causes severe inflammation and tissue damage. Apoptosis, on the other hand, is the controlled, highly regulated demolition of a cell. It is a genetically directed process executed by a family of specialized protease enzymes called caspases. These enzymes act like molecular scissors, systematically chopping up the cell's DNA and cytoskeleton. The cell's remains are packaged into neat little bubbles called apoptotic bodies. Neighboring immune cells, known as macrophages, then come along and quietly consume these bodies. Because the cell membrane never bursts open, absolutely no inflammation occurs. Now that we understand what apoptosis is, you might be wondering why a living organism would ever want its own cells to die. The truth is, programmed cell death is just as important as cell division for our survival. For example, when you were a developing embryo in the womb, your hands initially looked like solid paddles. It was apoptosis that caused the cells between your fingers to die, perfectly sculpting the individual fingers you have today. Beyond development, apoptosis is our body's primary defense mechanism against cancer and viral infections. If a cell detects that its DNA is mutated beyond repair, or if it realizes it has been hijacked by a virus, it will bravely trigger its own death to protect the rest of the body. This incredible defense mechanism leads us to ask how apoptosis happens so quickly once it starts. The answer lies in the mathematics of the caspase amplification cascade. Conceptually, a small initial signal triggers a massive downstream effect because enzymes can act on multiple targets repeatedly. In our formula, N sub E represents the total number of active executioner caspases, which are the enzymes that actually destroy the cell. N sub I represents the initial number of initiator caspases triggered by the death signal. Finally, F sub A represents the amplification factor, which is the number of executioner caspases that a single initiator caspase can activate. If the initial signal increases, the total destructive power increases proportionally. This mathematical relationship explains why apoptosis is an irreversible all-or-nothing response. Before we move on, let us see this amplification with a concrete example. Suppose a cell is exposed to a mild dose of ultraviolet radiation, causing a small amount of DNA damage. The cell decides to undergo apoptosis. We are given that the initial number of initiator caspases is 40. We also know from laboratory measurements that our amplification factor is 1,500. We need to calculate the total number of active executioner caspases that will be unleashed. We simply multiply the initial number of trigger enzymes by the amplification factor. Substituting our values, we multiply 40 by 1,500. The final answer is 60,000. Physically, this massive number of destructive enzymes will flood the cytoplasm, quickly overwhelming any survival signals and ensuring the cell is dismantled. Having calculated the massive output of this cascade, let us visualize exactly what this looks like inside the cell. At the top of the diagram, you can see a small cluster of initiator caspases, representing the initial biochemical signal. Notice how the arrows branch out exponentially as they move downward. Each initiator caspase activates multiple executioner caspases, represented by the red scissor icons. This visualizes our amplification factor. Finally, these executioner caspases target the structural components at the bottom, chopping up the DNA and cytoskeleton. This diagram beautifully illustrates why the apoptotic cascade is so efficient and deadly once the decision to die has been made. Now that we understand the execution mechanism, we must ask, how does the cell actually know when to trigger this cascade? There are two main pathways, and we will explore the extrinsic pathway first. Think of the extrinsic pathway like a self-destruct button located on the outside of a high-security building. The cell might think everything is fine, but a patrolling immune cell notices it has been compromised by a virus. On a molecular level, this button is the FAS receptor, or death receptor, on the cell membrane. An immune cell binds to it using the FAS ligand. This forms a structure inside the cell called the death-inducing signaling complex, or DISC. The DISC recruits and activates caspase 8, which acts as the spark that ignites the executioner caspases. Understanding this external trigger leads us to how scientists measure its effectiveness in the lab, especially when testing new cancer drugs. They do this by calculating the apoptotic index. Conceptually, this formula simply finds the percentage of cells in a population that are dying. In this equation, IA stands for the apoptotic index, which is a percentage. NA represents the number of cells actively undergoing apoptosis in a given tissue sample. NT represents the total number of cells in that same sample. By dividing the dying cells by the total cells and multiplying by 100, we get a clear percentage. If the value of NA increases while NT remains constant, the index increases, indicating a stronger apoptotic response to the external stimulus. Let us apply this formula to a clinical research scenario. A scientist is testing a new immunotherapy drug designed to stimulate the extrinsic apoptosis pathway in liver cancer cells. We are given a total counted population of 4,500 cells. The scientist observes that 810 cells are showing clear signs of apoptosis. We need to calculate the apoptotic index to determine the drug's efficacy. We place 810 in the numerator and 4,500 in the denominator. Dividing these gives us a decimal value of 0.18. Multiplying by 100 gives us our final answer of 18. Physically, this tells the scientist that the drug successfully triggered the extrinsic death pathway in 18% of the cancer cells, proving it is highly effective. We've seen how external threats are handled, but what happens if the danger comes from inside the cell, where the immune system cannot see it? For that, the cell relies on the intrinsic pathway. If the extrinsic pathway is an external button, the intrinsic pathway is an internal smoke detector. It senses severe internal stress, such as irreparable DNA damage from UV radiation. The central hub for this detector is the mitochondrion. Under severe stress, the mitochondrial membrane becomes permeable, leaking a protein called cytochrome C into the cytoplasm. Cytochrome C binds to a protein called APAF1, assembling into a massive wheel-like structure called the apoptosome. The apoptosome acts as a powerful engine that activates caspase 9, which then activates the executioner caspases. Knowing that mitochondrial leakage is fatal, we must explore how this critical event is regulated. The regulation of this leakage is literally a matter of life and death. controlled by the BCL2 family of proteins. This family is divided into two opposing teams. The pro-apoptotic proteins, like Bax and BAC, want to punch holes in the mitochondria to release cytochrome C. On the other side, the anti-apoptotic proteins, like BCL2, act as guards, plugging the holes and keeping the mitochondria intact. The fate of the cell depends entirely on the balance of power between these two teams. If a cell's DNA is severely damaged, it produces massive amounts of pro-apoptotic proteins, overwhelming the guards and triggering death. This biological tug-of-war can be perfectly described using mathematics. We can represent this balance using the protein concentration ratio formula. Conceptually, this formula compares the strength of the death signals against the survival signals. In this equation, R represents the survival ratio, which determines the ultimate fate of the cell. C-pro represents the total cellular concentration of pro-apoptotic proteins, which drive the cell toward death. C-anti represents the total cellular concentration of anti-apoptotic proteins, which protect the cell. If cellular stress increases, the concentration of pro-apoptotic proteins rises, causing the ratio R to increase. If the ratio becomes significantly greater than 1, the pro-apoptotic forces overwhelm the defenses, and the intrinsic apoptosis pathway is irreversibly triggered. Let us put this ratio to the test with a real-world scenario. Consider a skin cell exposed to intense sunlight, resulting in significant UV radiation damage. Biochemical analysis reveals that the concentration of pro-apoptotic proteins, specifically Bax, is 1,200 nanomolar. The cell is trying to survive by producing anti-apoptotic BCL2, measured at 400 nanomolar. We need to calculate the survival ratio to determine if the cell will live or die. We divide the death-promoting concentration of 1,200 by the survival-promoting concentration of 400. The final answer is 3. Because the ratio is 3, which is much greater than 1, there are 3 times as many death-promoting proteins. Physically, this means the anti-apoptotic guards are overwhelmed, and the cell will safely undergo apoptosis. Having calculated the cell's fate, let us look at the physical machinery of this internal pathway. On the left side of the diagram, you see the mitochondrion. Notice the pores in its membrane, allowing the red cytochrome C molecules to leak out into the cytoplasm. This leakage is the direct result of the pro-apoptotic proteins winning the tug-of-war we just calculated. As you follow the arrows to the center, you see cytochrome C binding with APAF1 to assemble the massive, wheel-like apoptosome. This apoptosome then activates caspase 9, which flows downstream to activate the executioner caspases, ultimately leading to cell death. This diagram perfectly captures how internal damage is translated into a controlled cellular demolition. We have covered a tremendous amount of ground today, so let us summarize the most important ideas. First, apoptosis is a highly controlled, genetically programmed cell demolition process driven by an amplifying cascade of caspase enzymes. Second, the extrinsic pathway acts as an external self-destruct button, triggered by immune cells binding to death receptors, which activates caspase 8. Third, the intrinsic pathway acts as an internal smoke detector, relying on the balance of BCL2 family proteins to release cytochrome C from mitochondria, activating caspase 9. Finally, whether triggered from the outside or the inside, both pathways converge on the executioner caspases, ensuring dangerous cells are safely removed. You now have a masterful understanding of programmed cell death pathways.